So it's with great pleasure that I get to introduce Charles Sawyers. Technically, he's a Hughes investigator and the head of the Human Oncology Program at Sloan-Kettering. But Charles' career is so incredibly distinct. I mean, I know very few people that have ever developed one drug. Charles, in his still growing, I mean, he's at the zenith of his career right now, actually gets the credit for developing two that have really impacted the lives of prostate cancer patients in hem-onc and now in prostate cancer with the approval last week of enzalutamide. So thank you for what you do. You're making a big difference.
Thanks. So Howard visited me in New York a few weeks ago, and he was sitting in my office and he said, "Charles, this is a really important talk. We want you to give a TED Talk." I didn't know what a TED Talk was. I went to the website. My wife knew about TED Talks. She made me watch 10 of them. So every TED Talk starts this way. "I'm Charles Sawyers. I develop cancer drugs." And then you go. So actually, Howard, it's 3 drugs. 2 for leukemia, 1 for prostate cancer, as of last week, all of which have been approved by the FDA. So I didn't dare put the life and times of my title on this one, but the question everyone asks me is, how do you do it? So I'll tell you my life and times. And it all goes back to here. So for those of you who are younger, this is what Facebook used to be. This was the freshman year medical school class at Hopkins in 1981. These 3 individuals were randomly assigned by the housing office to be roommates in Reed Hall in East Baltimore. The 3 of us came from liberal arts colleges where we were taught to ask questions, dig deep on problems, don't worry about memorizing facts, you can always look them up, and question authority. Needless to say, after the first week of medical school, we had become comrades in arms, rebelling against the fire hose of facts that were being shot at us each morning by the basic science lectures of first-year medical school. I think I'm actually very serious in saying that a lot of what I've accomplished, I think, gelled through this sort of spirit that began with these other 2 individuals. Now, I was a little bit different. I actually came to medical school as a history major. Knowing that I wanted to do science and become a doctor, I had no idea if I wanted to become a scientist. Jonathan and Bill had a little bit more scientific training, but after I failed a few biochemistry tests and they helped me through the first year, we were comrades. So the next key time in my life, I was 1 year old at the time this discovery was made, but this came to visit me in a real way when I was a resident at UC San Francisco in 1986. I was on the leukemia service. I was taking care of a 25-year-old fireman who had chronic myeloid leukemia, and he had received an allogeneic bone marrow transplant. He had a wife, young wife, really strapping young man, had an exercise bike in his room, all kinds of family coming in. And he had sailed through the conditioning regimen, and he'd gotten a matched related donor transplant, a curative therapy, known to be curative, but still in its infancy in terms of how to manage the complications. I took care of him through that month-long period of time until one day I came in and he had a big, bright red rash. His liver function tests had shot up overnight. He died within a week of acute graft versus host disease. That's when I started reading about CML, and I obviously noticed this literature from 25 years earlier, which was the description, the first molecular description of an alteration in a human cancer. You can see the arrow pointing to the short 22. Peter Knoll, when he saw this under the microscope, didn't have the benefit of the banding technologies that you see in this slide here. This is using what seems such crude technology to us today, but this is what allowed Janet Rowley to recognize that, in fact, this wasn't just a short 22, this was a reciprocal translocation of material from 9 to 22. So 1986, I'm thinking about this patient. I'm trying to decide what I want to do next as I finish my residency. And I read this paper from Owen Whitty, which in cartoon form made this important discovery, and that is that translocation breaks two genes and rejoins them and creates the B-seriable oncogene. His lab was the first to clone the full-length cDNA. I joined his lab 2 years later as a postdoctoral fellow and as a fellow in hematology oncology at UCLA to study this alteration, to see if it caused cancer, and in the remote recesses of my mind, thinking that perhaps it could be useful for therapy. I could never have imagined at that time how quickly that would come to pass. So you're all familiar with this paper from 2001 in which Brian Drucker and Moshe Talpaz and I reported the results of the phase 1 clinical trial of imatinib or Gleevec, showing remarkable response rates in patients with chronic phase chronic myeloid leukemia who had failed interferon, which was the current therapy, and were ineligible for transplant. This is now in the textbooks. But what the paper that you probably don't remember, which appeared right after this, is this one. And I actually think this is the most important discovery that we made, and that is this single agent, which inhibits just the B-seriable kinase in these patients, was effective in patients with blast crisis. Now, to give this prostate cancer audience some perspective on CML blast crisis, this is a lethal phase of the disease with a lifespan of a few months. It is considered hopeless. And the first patient I treated in the clinic at UCLA with imatinib who had blast crisis had a bone marrow full of blasts, as shown in the top left, Philadelphia chromosome on the bottom right, and the little arrows are showing lots of other genetic alterations. The genomes of blast crisis patients are just shredded. You can't imagine that a single agent could possibly have any activity. Yet 4 weeks later, this patient was in complete remission. The Philadelphia chromosome could not be detected. The only alteration we could still see was a little bit of a problem on chromosome 9. And here's the tally in that paper of response rates. Now, a complete response is just what I just showed you, complete remission. Those are pretty respectable numbers. I think any one of us would love to have a clinical result in any solid tumor early on in a clinical development. The larger numbers include partial responses. So this would be the entire waterfall plot that we're so used to looking for. So, I'm asking you, does anyone remember this paper? I think this is an incredible lesson because it says that you can get activity when you hit the right target, even in a genomically complex cancer. The reason you don't know this story is this slide. These patients all relapse very quickly. The numbers along the bottom are days. I'm showing you 1 year here. This was the most emotionally upsetting experience for the patients, for the physicians, all of us who were involved in the early phase of this trial. What did we do? We went to the lab. I formed a team, just like Liz said, 2 trainees in my lab, one a physician scientist who was working with me in the clinic, Neil Shaw, and one a PhD graduate student, Mercedes Gore. And we used a number of logical strategies to try to map down what was happening. And I'll never forget the day in early 2001 when Mercedes came into my office with the sequence traces from samples from patients on this Kaplan-Meier who were relapsing. So this was pre-whole exome, whole genome. This was PCR amplicons, painstakingly done by a single graduate student, looking for changes in the alphabet in patients who are relapsing. And I've given you the answer. It's those 6 out of 9 patients that we looked at had a typo. They had a T instead of a C at that one position. There was nothing else different that we could find at that point. That was an important letter change because it leads to an isoleucine at that position instead of a threonine. That's where things stood until John Currian, a structural biologist then at Rockefeller, now at University of California, Berkeley, someone who I did not know, someone who's interested in kinases as molecular motors, interested in understanding their structure through crystallography, published the first crystal structure of Abel bound to Gleevec. The left-hand panel shows you his structure. Abel is the Abel backbone is in green. The drug is in that gray cloud, and there's that threonine. He wrote an entire paragraph in his science paper about the importance of this threonine residue in forming a hydrogen bond with the drug deep in the ATP binding pocket. He knew very little, if anything, about CML. We had sequence traces showing that that threonine was now an isoleucine at relapse. One of my lessons to the young people is don't be afraid to cold call someone if you want to get some information. I called John up. He had no idea who I was. I had no idea who he was. Within 15 minutes, he sent me the PowerPoint slide on the right in which he had just plugged an isoleucine into his computer model, and it was absolutely clear that these patients were relapsing because the isoleucine can't provide the hydrogen donor for the hydrogen bond, and it's too bulky for Gleevec to fit into the binding pocket. The last sentence of our science paper says something to the effect that wouldn't it be a possibility to chemically modify Gleevec or come up with another compound that can accommodate the extra bulk of that isoleucine, and you could overcome resistance. And that's what we said about thinking, how could we get there? But Neil and Mercedes continued to sequence more patients, and the problem became much more complicated. Eventually, we and others demonstrated that there's over 50 different amino acid substitutions that occur at relapse and can cause resistance. So this is another version of the structure in which 13 of these different mutations are shown. The one that I just showed you is that purple ball up on the upper left of the structure. But you can see that the green ones and some of the pink and red ones are in places very distant from the drug. So fortunately, we had maintained this collaboration with John Currian's group, and every time we found a new mutation, we looked at it, and gradually a pattern started to emerge. So I'm going to turn the molecule, and I want you to see that this pink loop, you should visualize it coming out of the board at you, and you can also see that part of the drug is running behind that loop. This is just one of many lines of evidence that the reason imatinib binds to Abel is it so tightly is it has to get into this shape in order to bind. But the Abel protein is moving all the time. What these mutations do is impair its ability to get into this optimal shape. At least that's what we thought. That's what we wrote in our paper. That's what we talked about at meetings. And then the next cartoon shows this a little more graphically. So here the pink loop is now in red. And what you're going to see is that it's in the open conformation now. It's going to close. You can see Gleevec and purple binding only when it's closed. So what we put forth was the notion that is there a drug that could fit in the same binding pocket but fit in a way that it didn't care where the activation loop was? And for reasons that are too long to go into, we thought that such a drug might also inhibit another kinase called SRC, because SRC kind of looks like that same shape. And after speaking at one of the AACR annual meetings, someone raised their hand or called me afterwards, Francis Lee from Bristol Myers Squibb, and said, we think we might have such a compound. It's a SRC inhibitor. It's also an Abel inhibitor. We tested it on a panel. We have it in our inflammation program because we think it might be a good drug to inhibit T cells, but we'd like to give it to you to try against the CML mutants. It worked unbelievably well. Neil Shaw did those experiments and helped Moshe Talpaz and I lead this clinical trial, which was reported just 5 years after we first described the mutations. Dasatinib is now approved as frontline therapy for CML because when in head-to-head comparisons against imatinib, it's proven superior. So here's a sort of historical view of how that story unfolded. It took 25 years to go from the first morphologic description using crude chromosome visualization technology to 1985, the molecular revolution allowing the precise molecular identity of the driver mutation to be identified. Once that happened, though, things began to move quickly, and we had an approved drug. The bottom three historical points point out that once you know your target and you understand resistance, you can move even quicker. So I want you to keep these dates in mind because now I'm going to shift to enzalutamide. So in parallel with the studies of Gleevec resistance, we initiated a program to understand resistance in another cancer, and the cancer we chose was prostate cancer. Why did we choose prostate cancer? Well, it's an incredibly important problem. There are 5,000 new patients a year diagnosed with CML every year in the United States. There are 30,000 men who die of castration-resistant prostate cancer every year. I wanted to work on something that was a bigger public health issue. I also wanted to work on a different type of drug, in this case, hormone therapy. So using the strategies that we had applied with CML, but with some twists, we applied the same approach. The twist is we didn't have patient material that we could work with directly, so we used mouse models, some of which we derived, many of which we obtained through collaborations with some of you, in particular Bob Vasella's group at the University of Washington. And using transcriptome analysis of these tumors as they transited from a hormone-sensitive to a hormone-refractory stage and examining the data, we found this finding. I'm dating this array analysis roughly at 2002. The paper was published 2 years later because we had lots of functional studies to do, but I will never forget, again, that group meeting in which Charlie Chen, postdoc leading this work, showed this table in which the only gene that was consistently altered across 7 different xenograph panels that underwent this resistance transition was the androgen receptor. Now, to put this into some historical context, we actually were pretty disappointed with this result. We were looking for a kinase, we were looking for anything, something else that was going to drive these prostate cancers because they were androgen independent. It just didn't make sense that necessarily this would be the new driver of disease. But Derek Welsby, an MD PhD student in the lab, said we have to do the functional studies. He developed at that time what was nascent technology to do knockdown shRNA knockdowns, stable knockdowns in tumors in vivo, and showed that it was required for maintenance, and overexpression of the receptor was sufficient to cause resistance. So at this point, I've lived through Gleevec, I'm in the midst of dasatinib. I could not let this go and just write a Nature Medicine paper. We had to do something about it. I had a little bit of street credibility now with Novartis, who makes imatinib, and with BMS, who makes dasatinib. No take-up of this data to initiate a drug discovery program for a next-generation anti-androgen. So we formed an academic team. And here are the key people. Michael Jung is a synthetic chemist, senior professor at UCLA. His living is to make complex molecules to deal with this sort of simple translational thing. Initially, he thought might have been beneath him, but fortunately, he came around. Samedi Ouk was a postdoc in his lab who, very importantly, had spent a year in a pharmaceutical company in France after originally coming from Asia. And there he learned some medicinal chemistry, which was critical. And Charlie Chen and Derek, I mentioned earlier, were the biologists in my lab. And we set about looking for a compound that would work, work means inhibit the growth of prostate cancer cells when they expressed high levels of the androgen receptor and were resistant to bicalutamide. We first looked in vitro, obviously, but we knew we had something when we did this xenograph study in 2005, in which the waterfall plot shows that this RD series compound called RD162, as we named this series, shrunk tumors in all the mice we tested. Very exciting. It was licensed to Medivation, and they initiated a clinical development plan with the exact drug that we had tested in the mice. Howard Shear reported this data officially in February of this year. For those of you who don't know the nitty-gritty, this result was triggered by an interim analysis after the first 520 events of this roughly 1,200 patient study, in which the Data Safety Monitoring Committee decided to end the study early because of this survival advantage seen in the Medivation-treated patients. So I know you've also recently seen the paper published in its entirety in the New England Journal. A very proud moment, obviously. So here's the timeline for enzalutamide. The first data that I can really, trace back to saying the AR hypothesis is there emerged from those xenograft studies in 2002. We began that collaboration with Mike Jung within a matter of months. It took a while, but not an incredibly long time, to get to compounds that had in vivo activity in animals. It took roughly a year to get the licensing and then the IND processing done by Medivation. And first patient, first dose was at Memorial Sloan-Kettering under Howard's leadership in the summer of 2007. The first 6 patients, 3 on cohort 1, 3 on cohort 2, all had declines in their PSA level. We knew we had a drug. As you heard, it was approved last week. Why did it take 5 years to go from proof of concept, we knew we have a drug, to approval? In chronic myeloid leukemia, that timeline is 2 years. Why is it 2 years? We have a surrogate endpoint. We can do chromosome analysis of the bone marrow, a cytogenetic remission, or now a molecular remission, predicts for survival advantage. You can get a drug approved at a 12-month time point. We need this for prostate cancer. So I want to conclude by sharing some other data that's not from our lab, but I'm hearing talked about a lot at cancer meetings, and it's leading to some pessimism about targeted therapy. So this is a widely discussed New England Journal paper from a few months ago by the group at the Sanger Institute. It's a beautiful piece of work in which whole genome sequencing was conducted in a couple of kidney cancer patients, sampling different regions of the same tumor. For those of you who didn't see the paper, I urge you to look at it. I think it's one of the most important pieces of genomic work in the last several years. And on the bottom of this slide is a cartoon that depicts what the findings say. And that is, there's this concept of heterogeneity through branched evolution of mutations, even within a tumor that's in situ as a primary, with no treatment. This isn't Darwinian selection for drug resistance. This is just natural evolution of a tumor growing in the kidney, presumably with a hypoxic environment influencing it as well. And so the scary part of this, of course, is that the drugs that we're using to treat many of these mutations are, for example, hitting the PTEN pathway, are occurring in sub-branches of this tumor. And they will never get around this. BCR-ABL and CML, perhaps that's a trunk mutation. That's one of the blue line, and we can get away with it. If we were treating blast crisis with drugs that hit much later in these branches, perhaps we wouldn't have that success. So I'm hearing a lot of negativity about this, but I actually, like we heard earlier this morning, I'm an optimist as well as Jim Blair. And I want to share at the end this review article that I co-published with a colleague named Mike Glickman at Sloan-Kettering, who's an infectious disease scientist who works on drug resistance to tuberculosis. And at one of our annual Sloan-Kettering retreats, we happened to be on the podium back-to-back giving talks on TB and cancer, and we gave essentially the same talk. And so we decided that we should put our thoughts into words. I encourage you to read this, but I'll share one thought that I think comes out of this. It's stuff you know, but I think this brings it home. So these are cartoons comparing the response rate based on solid historical data with HIV on the top left and with tuberculosis on the bottom left. So the dotted line is the result with single agent therapy in both cases. You see partial responses and you see rapid acquisition of resistance. With HIV, when you add 2 or 3 drugs, heart therapy, 2 things happen. First of all, the slope of decline, the dark green line, is steeper. If you were to translate this to our waterfall plots, this would mean we would see waterfall plots in which more people are on the far right with tumors that are shrinking 100%. The second thing, of course, is that relapses occur much later or they don't occur at all. I know we can achieve this. We all talk about combinations, but we're not doing it fast enough. So my message to the young people here is let's not let this idea just be in review articles. Let's act on it. I've talked to the infectious disease specialists from the HIV era who did these clinical trials, and they had to get angry. I was talking with Ted Goldstein earlier, who's going to speak after me. He was asking me how important HIV activism was in making this happen, and it was important. HIV patients were handcuffing themselves to the desks of FDA officials demanding that viral load be used as a surrogate marker of disease and speed up endpoints. Clinicians were telling pharmaceutical companies, I'm not going to do a trial with your single agent compound. We need to do a combination trial with your sworn worst enemy's drug. Otherwise, we're not going to make any progress. So I'm here to tell you that I'm optimistic, but we've got to be more activist in making this happen. Thank you.
Questions for Dr. Sawyers? So what's going to be the 4th drug?
Well, we're working very hard on enzalutamide resistance, primarily from animal models, which, as we saw, were predictive. I don't know what that drug's going to look like yet, but I know the process that we're taking has a track record. So.
Yeah, I mean, just of note, Jonathan and I have dog-eared that original paper you did at UCLA showing amplification and mutation in AR. That is probably the best case for animal models that actually were instructive. What's wrong with the rest of them?
Well, I think some of them are more instructive than we give them credit for. I think you can see activity in an animal model, let's say lung cancer drug, and then you can do your trial in lung cancer and realize that you've now changed, you haven't selected your patient population correctly because that was an EGFR mutant lung cancer model, not a lung cancer. Mm-hmm. So I think that's now a well-known problem, but I certainly am not throwing animal models out of the table.
Either way. Thanks, Charles.
Okay.
Oops. You can't see us out here. I don't know how to turn
Is
this
that
on.
Pienta?
Yeah, it is. Thank
Name yourself and your institution,
Ken
please.
Pienta, University of Michigan. I appreciate the tour de force you just gave us, and thank you for what you've done for this field, Charles. I was wondering if you could comment, and maybe even Howard next to you could comment on this, your last bit about activism and getting companies actually to do the kind of combination trials that we all see as investigators are necessary. I know Howard Chairs spent a lot of time thinking about this too, because it seems like it's still a huge roadblock, and we haven't been able to do what AIDS and TB and other people have been able to do in the cancer field and in the prostate cancer field. So how do we, what's been the experience, I know, with PCF of trying to get this done, and are there actual physical steps we can make, discrete quantum steps that we can make as individuals to get this done besides chaining ourselves to desks?
I think one, which I tried to throw at you pretty hard, was we need a surrogate marker, maybe not for drug approval, but for clinical activity, so that a doublet or a triplet, the signal can be measured as quickly as possible. So you can know that it's going to work, and then you can, the lawyers and the business people can agree to the collaboration and do the registration trial. So I think an early read for real activity is one of the most important motivators to get people to the table.
So what's your best guess where that marker is going to be in prostate?
I mean, it's going to sound silly, but I think big PSA responses that are sustained for 12 weeks, like we've seen with abiraterone and enzalutamide, they seem to be reasonably predictive. I don't know that we should throw that out with the bathwater. Circulating tumor cells, circulating DNA, actually, circulating tumor DNA from work that I've seen from the Vogelstein group is so quantitative over a broad dynamic range that I would love to see that move forward. That seems to me like the BCR-able molecular response that's now du jour in decision-making in CML.
Well, we do have one effort based on the results of Mary Ellen Taplin that she reported at ASCO, showing a large number of near-complete responses and some complete path responses in the neoadjuvant setting with Lupron plus abiraterone. Under Phil Kantoff and Chris Sweeney's leadership, we've opened a new endpoint discovery program looking at lots of different trials and what people have found, and trying to, because the further you get back in the natural history of the disease, the earlier you go, the more complicated it is, because it's not leukemia. The good news is these patients live for a long time. The bad news is survival is the only approval.
Yeah, but I think going early is a really important point. I think going early is chronic phase CML, CRPC is blast crisis.
Exactly. Okay, Don?
Vogelstein, as you know, has just said that he thinks it's unethical to do a single drug for the very reasons you're saying. I don't know if that could be developed into an understanding, but he says the same thing, that you got to go early and you got to go multiple. To go single ain't going to work. Now, if you had some way to say that this is a good combination, and that's what you're really saying, you need a marker for that.
Well, you need a marker for response, but I think there's plenty of good science arguing for certain combinations. It's just hard to get the players off the dime. And Bert's right. I think it is unethical, but we as a community of clinical trialists have to say, we're not going to do your trial that way.
Exactly.
So do you, Howard? You know a little bit about the overview of the Congress and everything, the lawyers and the legal aspect. What would you suggest?
I mean, I spent a long half day with the CEO of Aragon a few weeks ago, and I think the answer is in small, hungry companies. I think trying to get a large company to do a combination trial, I don't know, Howard Scher, you might want to, I mean, this is really complicated. The small companies that really want the answer are more willing to work together. Unfortunately, we lost, and Telekine got taken out. But that was the eve of doing some really interesting combo studies with PI3 kinase inhibitors and AR antagonists. So we're working on it. Thank you, Charles. That was brilliant.